All right, so today's lecture is going to be on the four different types of tissues. So the four different types of tissues are going to be epithelial tissue, connective tissue, muscle tissue, and neural tissue. So again, remember, cells come together, a group of cells come together and make tissues. And those tissues come in these four flavors: epithelium, connective, neural, and muscle. Now, what we covered in earlier weeks, we did discuss muscle because that goes pretty quick. We said there's three types of muscle: skeletal, smooth, and cardiac. Skeletal, smooth, and cardiac. And we briefly discussed some of their characteristics. In today's lecture, we will show you what those three look like. In the neural tissue, we said under the nervous tissue, I don't like to say nervous, so you'll hear me say neural or nerve, but neural tissue, we have two types of cells that fall under neural. That would be the neuron, that is the main one, and then there are neuroglial cells. There are about six. But we are primarily interested right now in understanding the main parts to a neuron. And you will see me use my hand and my forearm as a classical example to understanding what a neuron is. Having the fingers of your palm represent the dendrites, the palm of your hand represent the cell body or the soma, and then the forearm representing the axon. Those are the three main parts: dendrite, cell body, and axon. The fingers being the dendrites, which means dendra branches, the palm being the cell body or the soma, and then the forearm being the axon. There's a few more moving parts, but those are the biggies. What we need to differentiate now is the main differences between epithelium and connective tissue. And one of the big differences that I'll show you under the microscope, when we look at epithelium, it's a bunch of cells closely packed together with very little extracellular matrix. Whereas connective tissue is some cells here and there with lots of extracellular matrix. That's the big difference in terms of appearance. And we'll show you what they look like under the microscope. So each tissue is a little bit different. Their structure and function is different, which makes a whole lot of sense. If their structure and pattern or form is different, then so are their functions. Cells come together to make tissues. Cells come together with different types of junctions to form tissues, like gap junctions, tight junctions, desmosomes, hemidesmosomes, adherent junctions. You'll learn a little bit more about that in your lecture. But generally speaking, cells come together by junctions to make tissues, and then tissues come together to make organs. When we study tissues, we call that histology. So when we look at microscopic slides, you'll see histology slides because we're looking at different types of tissues. When we look at epithelium first, epithelium is the type of tissue that is going to line the body and cover the body. It covers the external surface. So when we think about, hey, what do we know of that covers the entire external surface of the body, you should be thinking the skin. And yes, the outer covering is called the epidermis. And if we go a little bit deeper, that is the dermis. If you remember back to what we covered in the skin model, which is on another video, if you have not subscribed yet on YouTube, make sure you go to Nutrition Chirodoc. That's Nutrition Chirodoc. Chiro is CHIRO, doc, DOC. Nutrition Chirodoc. Subscribe so you get all of these updates on the videos. One of them is on the skin, on the integumentary system. The skin, the epidermis, has those 5 superficial layers. And we said Cher loves getting skin Botox. Just take the first letter of each of those words in that sentence. Cher loves getting skin Botox, and you can figure out the 5 stratum layers. So Cher, the C, is stratum corneum. Loves is stratum lucidum. Getting, share loves getting. Stratum granulosum, skin, stratum spinosum, Botox, stratum basal layer, or germinativum. Those are the 5 layers of epithelium in the epidermis. And then deep to the epidermis is the dermis. That's where we had a lot of our accessory structures. We have the sebaceous gland and sudoriferous gland and the erector pili muscles, right? And there were some neural structures in there. And then you go deeper into the hypodermis or the subcutaneous, and that's where you had the adipose, which we'll cover today, which is under connective tissue. So epithelium lines the external surface of the body, and we'll also see it lining body cavities and tubules. Tubules, you'll hear tubules within the kidneys. The kidneys have, especially the functional unit of the kidney that we call the nephron, has tubules. They're just called the proximal and distal convoluted tubules. But epithelium is also located in tubular systems, meaning within the body, anything that actually looks like a tube. Arteries and veins look like long tubes. The esophagus and trachea look like long tubes. The urinary tract is a tubular tract. The reproductive tract is tubular. The digestive tract is tubular. So really anything that looks like a long hose or a long tube with a hollowed out lumen or cavity in the center is going to be lined with epithelial tissue. Glands are also classified as epithelium. Their primary functions is protection. I mean, if you think of the skin is going to protect us against excessive friction, any type of bacterial or viral type of invasion, any type of chemical damage. The skin is that first line of defense of our immune system to protect us. There's also cilia, which are these little hair-like or finger-like structures that can come off of epithelium. So we call it ciliated epithelium. Just think of cilia as like, how about like the bristles on a broom, right? That would be like cilia. And it can sweep, right? It can sweep dust or foreign particles away. So ciliated epithelium, or these little hairs that come off the tissue, we'll see them in the trachea, in the respiratory tract, and they're constantly beating, they're moving or beating upward from inferior to superior to try and sweep and move every particles and dust away from the deeper part of the lungs and move them up and out of your respiratory system. So it can allow you to go like this and just cough or clear it and get things out of the respiratory tract. Cilia are very prone to damage as well, especially from smoke. We don't want those cilia burning off. We don't want them becoming paralyzed. We want them functional and constantly beating superiorly to clear out any debris. When we look at ciliated epithelium within that type of tissue, we'll see cells that are called goblet cells. And goblet cells produce mucus. So that mucus that's produced by those goblet cells are going to be hanging out and sitting on the cilia. So it acts as like an adhesive and a glue for all that debris and dust and particles to stick to. So ciliated epithelium is very, very important. Epithelium is used for absorption, filtration, excretion, secretion. These things, especially filtration, excretion, and secretion, we're going to see these a lot in the kidneys because this happens to be the function of the kidneys. The kidneys are filtering, they're secreting things, and they're also excreting things from the body. Excreting is getting rid of. Absorption is like a sponge, right? If something is absorbing like a tissue or a sponge, we're going to find absorption function very, very big throughout the GI tract. GI means gastrointestinal tract, meaning stomach and intestines, the GI tract, gastrointestinal. So we're going to see epithelium throughout the entire, through the mouth, through the throat and the stomach, the esophagus, the stomach, small intestine, the duodenum, jejunum, the ileum, the colon, all parts of the intestines, all parts of the colon. So epithelium is important, and sometimes students get confused with this concept. So when we think of epithelium, we're thinking of this covering concept where it's outside of the body. But if you think of the digestive tract, that is a tube within a tube, right? It's a tube within your body, another tube. So the digestive tract is really considered to be outside of your body, even though it's inside, from the concept that things do not really become part of your cells and tissues until you absorb them. So if you eat a healthy breakfast and it's in your esophagus and now it's in your stomach, that's still considered outside your body. It's until you digest it and break it down with digestive enzymes, then it pulls it deeper into the body. So if you swallow or inhale a quarter, it's easy to know that that's not truly inside your body, although you think it is because you don't see it anymore, but you end up pooping that quarter out a day later because it has not been digested and assimilated into your tissues. So what's inside the lumen of the esophagus and the stomach and the intestines, that is still considered outside of the body. All right. So epithelium is found throughout that entire tubular system. Even though it's in your body, it's still considered outside. All right. Just keep that concept in mind. Also, sensory reception is really big. So epithelium that covers the body's surface contains a lot of sensory receptors that will signal into the neural system. We already talked about cilia. Those are those little slender-like extensions. You've already seen them when we covered the cell, where you had these finger-like extensions that can help propel and move the cell around. It helps to also move secretions and fluid across the cell's surface. Microvilli are much, much smaller than cilia. They're microvilli. They're very, very small hair-like projections that are really used for absorption. So we're going to see microvilli throughout the digestive tract as well. Now let's get into epithelium in terms of how do we identify it? How do we name it? Because essentially, this is a big part of what students need to be able to do for their exams, whether it's lecture or lab, to understand how we identify epithelium. So simply speaking, just to give you a quick visual in your head, I said if you look on a kitchen floor or basement floor, usually there's tiles, or a classroom, a traditional classroom setting, there's usually tiles on the floor. If you could think of tiles as being, each tile is a cell and they're held closely together with very little space between them, that's epithelium under a microscope. It's lots of cells held closely together with very little extracellular matrix, with little stuff around it. Extracellular matrix means extracellular, the stuff outside of the cell. Intracellular means the stuff within the cell. So extracellular matrix is the stuff around the cell. Epithelium has lots of cells closely packed together, very little extracellular matrix. So once we see, hey, there's a bunch of cells held closely together, we know it's going to be epithelium. We then look at it and ask ourselves two questions. How many layers of cells do we see? How many layers? If we see one layer, we call it simple epithelium. If we see multiple layers, we call it stratified. And we're naming it according to the apical layer, meaning the layer that you see at the very, very top, the apex, or it's the number of layers. If we see a lumen, like a tube or a hose, we're looking at the layer that is closest to the lumen. Okay, that will make sense in a little bit when I show you the visual. So, first question, how many layers? One or more than one? If it's one, the first name of the epithelium is simple. And if it's more than one layer, we call it stratified. And then we have to ask ourselves, hey, what is the shape of the cell? Is it, does it look like a square or a cube? Then we call it cuboidal. Does it look like a rectangle or a column or a building? Then we call it a columnar. Does it look like a flat, irregular shaped cell? We call it a squamous cell. So we need to know the number of layers and the shape. Let's make some sense of this. So when we look at the top left here, where it says simple squamous, and then we look down below it, stratified squamous. So here is the apical surface, here's the top, or here's the top. And we can see here that this is just one cell layer thick. The bottom is the basement membrane. So this is just one cell layer. If we look down below, here's the apical surface. We see 1, 2, 3, 4, like, 3 to 4 different layers of these flattened out pancake, irregular shaped cells. So this is stratified squamous. We're not looking at the layer on the bottom. We're looking at the apical, the stuff that's on top. Okay. So then we look at the simple cuboidal. And when we look at simple cuboidal compared to stratified cuboidal, well, this is pretty obvious, right? We look at these. These are one cell layer thick of square cube-shaped cells. Then we look down below it and we see more than one layer. These are multiple layers of cubed-shaped cells. So this is simple cuboidal. This is stratified cuboidal. So we would call this simple cuboidal epithelium. We'd call this stratified cuboidal epithelium. We'd call this simple squamous epithelium. We would call this stratified squamous epithelium, right? Okay, then we look at these. Okay, well, this is obviously flat irregular, that is squamous. This is cube shaped, that's cuboidal. These look like the rectangles. These are column shaped. So these are columnar. And these two, although they're taller, it's one cell layer thick. So it's simple columnar. Something to know, FYI, there is no stratified columnar. There's no true stratified columnar. There's pseudo-stratified. Pseudo means false. So when we look at this, although it looks like it's multiple layers, your eyes are being fooled. They're being tricked. If you look at simple cuboidal, you see how all the nuclei are nice and even. If you look at simple columnar, you see how all the nuclei are pretty much at one level. Do you look at stratified cuboidal, you see that the nucleus are all right in the center. Even with squamous, they're all in the center. But when you look at pseudo-stratified, one nucleus is up here, another one's at the bottom, another one here, another one here. They're at multiple levels, but they're still only one cell layer thick in height. They're just different sizes of columnar. So they're not true stratified columnar. They call it pseudo stratified. And we'll see these quite a bit in the respiratory tract, sometimes even in the digestive tract. Okay. Transitional. How do I explain transitional? Okay, think of transitional. Imagine you have a balloon, and before the balloon is inflated, you make a circle with a Sharpie pen. Okay. Then you blow up the balloon. Now, if you made the circle the size of a dime when it was deflated, and then you blow up the balloon so that the balloon gets bigger and bigger and bigger, wouldn't you agree that the shape of that circle that you once drew, that was the size of a dime, becomes stretched out and can change its shape? Of course. Transitional is exactly that, where the cells have a variety of shapes depending on the organ, like the urinary bladder, for example, or the urethra or the ureters, Parts of the urinary tract, depending on whether or not the urinary bladder is filled with urine or not, that organ, that balloon can be inflated if it has lots of urine in it, or deflated if you've already emptied your bladder. Based on the stretching phenomenon that can take place, the cells can alter in their shape and size. Some look cubed, some look stretched, some look squamous, some look cuboidal. So if you see a mishmash, that's transitional. So we have simple epithelium and stratified epithelium. Simple epithelium consists of just one cell layer. Stratified epithelium consists of two or more layers. Now that we've identified the number of layers, you then go shape: squamous, cuboidal, or columnar. The squamous is the irregular, flat-shaped cell. Cuboidal is the square or the box or the cube-shaped, and columnar look like the columns. Their functions are slightly different because if their structure is different, their function is going to be a little bit different. Squamous is used for protection, secretion, and diffusion. The word diffusion is going to be associated with like particles or ions, sodium and potassium can diffuse through a membrane, right? It can pass through. Diffusion could be associated with gases like oxygen and carbon dioxide can diffuse from one side to the other, right? Like when you breathe in oxygen and then your body gives off carbon dioxide, those gases have to diffuse from cells to tissues, right? It has to go from a red blood cell, the oxygen, to the tissue to bring the oxygen to the other cells and tissues of the body. And then they're going to give off carbon dioxide, and we too have to get rid of that. So that's where diffusion. Protection, you understand, and secretion is going to be releasing something from the cell. So we're going to see squamous cells. If it's, think of the skin, right? I mean, the epidermis. Imagine it was just simple squamous. If you only had one cell layer of protection on the skin and you have nails on your fingers and you had an itch and you were to scratch yourself, those squamous cells would end up under your fingertips and you would bleed instantly because one scratch on your skin and you would remove all those cells. So we have to have multiple stratified layers in the epidermis, especially in the epidermis. You have to have stratified squamous epithelium in that top layer to protect ourselves. Cuboidal. These are going to be found in the kidneys where the tubules are. So when we get to some of the histology slides, you're going to see small little tubes with either simple cuboidal or stratified cuboidal going around it. But anywhere we see tubules, especially around the kidneys, at the proximal and distal convoluted tubules, we're going to see simple or stratified cuboidal. So you can, when you have time, you can kind of Google nephron, 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 and you'll be able to see the proximal and distal convoluted tubules. They look like little tubes, and they're going to be lined with cuboidal cells. So they're going to be used for absorption and secretion. And then we have columnar cells that will show you, we'll see them in the respiratory tract and even in the digestive tract. Okay, and again, we're looking at either the top surface, the apical surface, or at the level of the lumen. All right, we talked about stratified epithelium, but when we talked about columnar, we said there's only pseudo-stratified. There is no true stratified, only a pseudo-stratified in terms of columnar cells. The simple columnar epithelium, it's only one cell layer. We said that the nuclei can vary at different levels. When you see the nuclei at different levels, we don't call it a true simple unless the nuclei of columnar cells are all even. They're all at one level. Then we can say it's simple columnar. But when we see the nuclei at different levels, as I showed you before here, then we say it is not simple columnar. These are all even nuclei. When they're scattered at different levels, they're stratified columnar. Okay. So it's false, right? Pseudo is a false appearance of being stratified. And often when we see columnar cells, they're ciliated. We're going to see little cilia on top. Transitional, it looks like squamous on top, and then sometimes you see cuboidal deep to it. That's because the organ is being stretched. So they're found in the urinary system, especially the urinary bladder, the ureters, parts of the urethra, because that can expand, that can get larger, it can swell up, and then it can deflate. Just think of the balloon for transitional. So simple epithelium, we have simple squamous, simple cuboidal, simple columnar, and then for stratified, stratified squamous, stratified cuboidal, stratified columnar. Then you have those two little subspecialties, the pseudo-stratified columnar, and usually when you have the pseudo-stratified columnar, at the end of the columnar cells, you may see cilia. That would be normal to see. Not unusual, very normal to see cilia associated with columnar cells. Not with cuboidal, not with squamous, only with columnar. Transitional is with the urinary system, the urinary bladder, and the ureters. Okay, now we're finished with epithelium, right? So now we said, okay, a bunch of cells held closely together with very little extracellular matrix. Very, very little extracellular matrix. Now, connective tissue is the opposite. Connective tissue is when we're dealing with cells that are scattered, not close together, they're far apart, they're widely spaced, and there's lots of extracellular matrix. The matrix is made up of protein fibers and ground substance and liquids and gels and protein. That is the substance that goes around the cell. So connective tissue is also the most abundant tissue of the body, and it's also quite variable. So some of the functions of connective tissue is that it connects other organs together. We'll see certain type of tissue that can do that, like reticular tissue. It connects epithelium to the rest of the body. You'll see that connective tissue acts as like a part of a basement membrane in which epithelium can connect to it. So that's kind of neat. We'll show you that. It gives support and protection. In terms of support and protection, not just physical support and protection, but immune. And that's because when we talk about connective tissue, we have to bring in blood, not just red blood or red blood cells, but white blood cells. That is the immune portion, the WBCs, the leukocytes. So in a previous video that I made, we talked about white blood cells, right? We showed you the plaque of the blood where it had RBCs and then it had WBCs. We talked about the granular sites and the agranular sites. And the three granular sites were basophil, eosinophil, and neutrophil. Those had granules in the cytoplasm, which is why they were called granular sites. Those granules degranulate and release these potent, strong enzymes to destroy whatever pathogen they happen to be attacking. The agranular sites are the monocyte and lymphocyte. We said that when you look at the monocyte, that has a large cell. It's a large cell that contains a large nucleus that looks like a kidney bean. Whereas the lymphocyte is a nice round cell where the nucleus is just about as large as the cell itself. When the lymphocyte count is high on a blood test, we said that's associated with viral infections. And you know, you have T lymphocytes, B lymphocytes. When the granular sites are high in count, like the neutrophil, those are high with bacterial infections. When the basophil count is high, that's usually seen with allergies because basophils release histamine. And histamines are involved with allergies. And eosinophils are associated with allergies and parasitic infections. Eosinophils are associated with allergies because eosinophils release histamine ACE. So while the basophil is making histamine, the eosinophil is making histamine ACE, and anything that ends in ACE is an enzyme. That enzyme breaks down histamine. So they kind of work together. So those are the different types of white blood cells, and you're thinking immune protection. So you had basophil, eosinophil, and neutrophil. Those were granular. The other differences was that the eosinophil was a bilobe nucleus. The neutrophil had 3 to 5 lobes to the nucleus. And the basophil had that S-shaped nucleus with the very dark blue staining granules that typically obscured the nucleus and was hard to see. Whereas the eosinophil had those large red grainy granules. The neutrophil had much smaller pinkish reddish granules, but the eosinophil is much larger granules. In terms of storage of energy and heat production, that's adipose. That's fat. So when you look at the skin model, on the top was the epidermis, below it was the dermis. Remember, the epidermis had those 5 layers of epithelium. And then the dermis had those 2 layers, which was the reticular layer and the papillary layer. And then below the dermis is the subcutaneous. And in the subcutaneous is where we have fat or adipose. The other function of blood is movement and transport of materials. That's how things are transported and moved through the body. It goes through blood, which is in your blood vessels. That's the transport system. All right, now that we've talked about some of the basic functions of connective tissue, we have to look at some of the categories that fall under it. So one of the categories you'll see here is called connective tissue proper, and you have loose connective tissue and dense connective tissue. So you have loose connective tissue. We have 3 categories under it. We have dense connective tissue. So we'll talk about dense and loose. Then we have fluid connective tissue, which is blood and lymph. Then we have supporting connective tissue, which is cartilage. We'll talk about hyaline, elastic, and fibrocartilage. Under support, we also have bone. Okay, so for supporting, you've got cartilage, and then we have bone. And then we did a lecture on the osteon not too long ago that covered all the different parts of the bone that you need to go over. And then we'll talk a little bit about whether or not certain tissues have a good blood supply or they don't have a good blood supply, meaning are they vascular or avascular? And then we'll get into muscle and nerve. So let's go back to connective tissue. Again, there's loose connective tissue, dense connective tissue, fluid connective tissue, and then supporting connective tissue. So let's look at the loose connective tissue first. There's areolar, adipose, and reticular. And again, on the there will be a PowerPoint presentation, and I have a video of this that only goes over the histology slides, showing you areolar, showing your adipose, showing you reticular. We go through all of these on a separate video. So the loose connective tissue proper, there's areolar. Now, areolar is what happens if you were to pinch your forearm and pinch your skin, you can separate your skin from the deeper muscle. So that's areolar connective tissue. That's the padding that separates your skin from whatever lies deeper to it. And it's made up of elastin and collagen fibers. These are different types of protein fibers. Whenever you see elastin or elastic, you're thinking elasticity, the ability of something to stretch and rebound back to its original size and shape. Collagen, when you see collagen, you're thinking about a protein that's more designed for strength and stability and structure. Okay. Adipose or adipocyte, those are fat cells. Fat cells tend to store energy, they store sugar. We just call that triglycerides. And they store toxins. Adipose stores toxins. You don't want to consume food dye number 8 and red food dye, and then foods with carcinogens or pesticides and fungicides on them, and put all of these toxins in your body and have your neural system exposed to it. It'll kill it, it'll destroy it. So it knows to, hey, let's put it in storage, let's put them in fat cells for a while, and little by little by little, we will slowly release them into circulation. And now that it's in blood circulation, we'll send it to the liver to detoxify it. And the liver needs to be properly nourished to be able to detoxify. And if you were to research and look into liver detox phase 1, phase 2, there's a nice picture and illustration that shows you all of the nutrients needed for the liver to support an individual who's trying to detoxify themselves. When you consume toxins, that's pretty dangerous to begin with, but having the body detoxify to try and get rid of those toxins is just as dangerous. In fact, can be more dangerous if the liver lacks the appropriate nutrition. There's vitamins, there's minerals, and lots of antioxidants that the liver needs to properly do that efficiently. So adipose is very, very important. Again, you saw that in the subcutaneous of the skin. This is why the skin model is such an important model, because it covers epithelium and connective and muscle and nerve, as you'll see at the end of this PowerPoint. Reticular is also considered loose. It is really a meshwork of fibers that help produce the framework of the organ support system. Now, dense connective tissue, we have dense regular and dense irregular. And you'll see these on the slides comparing side to side. When you look at dense regular compared to dense irregular, dense regular is when you have collagen fibers, which are these protein fibers that are perfectly parallel in alignment with one another. They are densely packed collagen fibers. When you look at muscles and tendons and ligaments, you'll see that they are arranged in such a way that they are closely and tightly packed. The collagen fibers are moving tightly packed in the same direction in parallel form. That would be dense regular connective tissue. But dense irregular connective tissue is when those collagen fibers are arranged more loosely and not all tightly packed together, but they are arranged in a very irregular type of pattern. And you know that when you look at muscles and tendons, you'll look at the fibers and they're all in alignment. Some may be horizontal, some fibers may be vertical, or some fibers may be in a diagonal, but they're all moving in the same direction. When we look at the skin and the dermis, when you get massaged, a massage therapist doesn't just manipulate the skin or soft tissue in one direction, like up and down, left and right. They can move in circular patterns. There's no direction on your skin that says, okay, you can only move this way. A massage therapist can manipulate the skin and soft tissue in a variety of directions because those collagen fibers are arranged in a variety of ways. Elastic connective tissue, these are found in certain ligaments, especially bones or joints that have lots of flexibility to them, like the spine has a lot of flexibility. It can move in a variety of ways, but the spine is pretty stable. So when you have bones and joints that are stable to begin with due to their bony architecture, but they need to flex and extend and rotate, we're going to need ligaments that attach to them that allow for that elasticity. You need some collagen as well with those fibers and that type of ligament, but not as much as you would need elastic for the elasticity. For example, if you were to look at the knee is a pretty large joint, but it's very unstable based on its bony articular support. So the knee needs elastic connective tissue because it's such a flexible joint. But there we need lots of collagen for stability and support of the knee because it's such an unstable joint. Then we look at the fluid connective tissue where we have blood and lymph. Last week, we spent lots of time going over the blood. We spoke about the red blood cells and white blood cells and platelets. So make sure you review that other video on YouTube or that we have that's associated that shows the plaque, that shows you the red blood cell, the erythrocyte, the different types of white blood cells, which were granular and agranular. And then you had the plasma, which was the extracellular matrix, right? This is why blood is connective tissue because you have cells scattered far apart with lots of extracellular matrix. In blood, the ECM or the extracellular matrix is referred to as plasma. And then you had platelets, those little triangular star-shaped formed elements in the plasma that are involved in clotting. Then there's lymph, which is interstitial fluid that's found between cells of the human body. With supporting connective tissue, we have cartilage. Now, cartilage, as well as ligaments, have a very poor blood supply. They're avascular. Cartilage is avascular. Vascular means blood vessel. You put the A in front of it, it means without. Which means that cartilage between the knees and cartilage between the hips, right? Wherever you have two bones that come together that form a joint, we're going to have cartilage. And the movable joints, the freely movable joints, have hyaline cartilage. Like the shoulder joint has hyaline cartilage, the hip joint, the elbow joint. Okay, you're all going to find hyaline cartilage where you have freely movable bones. Elastic cartilage is what we're going to have in the nose and the ear. Right? If you take your ear and you scrunch it, you let go, it rebounds back. If you take your nose and you move the bridge a little bit to the left, it'll bounce back to the right. Elastic is also found around the lungs as well. Fibrocartilage, on the other hand, this is the strongest cartilage of the body. Your intervertebral discs are fibrocartilage. The intervertebral disc is fibrocartilage. It's a strong cartilage. So the discs. The pubic symphysis, where your two pubic bones come together in the front, there is a vertical disc that's there. Not horizontal like the discs between your vertebrae, the intervertebral discs. This one is vertical. It runs up and down. That's called the pubic symphysis. So again, cartilage is pretty much avascular, has no blood supply. So it's going to get all of its nutrients through diffusion from adjacent structures that lie around it. Muscles are vascular. Tendons have a little bit less blood supply, not as good. Ligaments also very, very poor. So when you talk about these tissues being damaged, if you stretch a muscle beyond its anatomical limit, that's called a strain. It can heal. It may take a little time, but it'll heal because it's got pretty good blood supply. Tendons have a little bit poorer blood supply, so it'll take a little bit longer. Ligaments, even worse. So it takes much, much longer for that to heal. And cartilage, the worst, horrible. So it gets its nourishment from all the adjacent structures that are next to it through diffusion and through the synovial fluid, which is the liquid within the joint. That helps to nourish and bring some nutrients to the cartilage cells themselves. We spoke about bone. Now, when we talk about bone, remember there's spongy bone and compact bone. Spongy bone and compact bone look very, very different. Remember the appearance of the osteon and be familiar with all these structures. Make sure you go back to my older video that discusses the osteon, which has all of those concentric lamellae, and it's got the central canal or Haversian canal that contains the blood vessel. We said that osteoblasts form the matrix of bone, but when the matrix and all those calcium deposits harden, the osteoblast gets stuck in a cavity called a lacuna, and now it becomes a mature cell called an osteocyte. That is a mature cell. It does not produce any matrix. It just helps to maintain the bone's integrity. The osteoclast works opposite of the osteoblast, where the osteoblast is building bone. The osteoclast breaks it down. It dissolves the bone matrix. We showed you in an earlier video the difference between the concentric lamellae forming those rings around the central canal compared to the circumferential lamellae, which are remnants of older lamellae. Okay, we'll show you some of of older osteons, not the circumferential, the interstitial, my bad. The interstitial lamellae, those are remnants of older osteons, and we'll show you what they look like in just a second. The canaliculi are those jellyfish finger-like extensions off of the osteocytes. And let's see if I can just show you pretty quickly here. So if this is a bone here, this is compact. So you can see all of these osteons here. This is an entire osteon. These are the concentric rings. Okay. Each of those rings is a lamellae, a concentric lamellae. The periosteum is the outer part, just like bluish grayish. That is the periosteum of the bone. You can see down here on the bottom, periosteum. But the inner portion, that is the endosteum. That is spongy bone. Spongy is on the inside. It looks like a very porous sponge. On the periphery out here, this is the compact bone. And again, we showed you this video. There's another video that goes through the osteon and all its moving parts. Make sure you know the osteon, the concentric lamellae. Number 5 out here, that was the circumferential, right? The circumferential lamellae, that goes outside the circumference of the entire bone. That's the circumferential, not to be confused with concentric lamellae or even the number 10 here, the interstitial lamellae. Those are between the osteons. Those are the remnants of what was or used to be an osteon until the osteoclasts broke those down. Okay, you'll see that here is spongy bone, number 1. Spongy bone is here because this is the endosteum region. And then all the way on the outside, number 3, that's your periosteum. And then you have number 9 over here, that's your central canal or Haversian canal. And that is where your artery, vein, lymph, and nerve travel. Okay, so make sure you review that. So connective tissue is highly vascular with the exception of cartilage, poor, ligaments, poor, tendons have some, but not as much as muscles. So muscles have the most, tendons come in second place. Third place would be ligaments, and then last would be cartilage. Very, very poor. When we cover muscle, okay, so that's the end of connective tissue. So we did epithelium, we did connective. Now the 3 types of muscle are skeletal, cardiac, and smooth. Skeletal is the only one that's voluntary. Cardiac and smooth is involuntary. Skeletal and cardiac are both striated. We'll look and see on the microscope, they have striations to them. Smooth looks smooth. They look spindle shaped. No striations. So here is skeletal. If we look at skeletal and we look at cardiac and I bounce back and forth, you can see the way the light bounces through here. It is striated. You could see those lines. You could see light, dark, light, dark, light, dark. These two, which are striated, looks very different than this one. This one is smooth. There's no striations. These two have obvious striations. Smooth has no striations. So skeletal is striated, and it's going to function to move the body because this is skeletal muscle. It attaches to the skeleton. Anything that attaches to the bone is going to be skeletal muscle. And when these contract and there's tension that's generated here, these muscles can shorten, they can lengthen, or they can remain the same length. That's where we will learn when we get more into muscles. We will learn the terminology concentric, eccentric, and isometric for different types of muscular contraction, which means tension that is being generated within the muscle. When the origin and insertion shorten, it's concentric. When origin and insertion of the muscle move further away, it's eccentric. And when there's tension, but there's no change in the joint angle, we call it isometric. Cardiac is striated, but it's highly branched. If you look at the difference between skeletal and cardiac is highly branched. You can see all the different branches coming off. And another characteristic here, it's intercalated discs. These lines, these dark red lines that run in the opposite direction of all of these fibers, these are called intercalated discs. And all intercalated discs is when you take one cardiomyocyte, which is one heart cell, and another cardiomyocyte, another heart muscle cell, and you bring them together. It's where the two fuse. It's where they come together. Remember, cells come together to make tissues. So this intercalated disc is really when we separate them and look inside, they're gap junctions. Gap junctions. Remember, gap junctions are used for fast, rapid forms of communication between cells, especially muscle and nerve cells. So right here, deep within the intercalated disc, if we were to separate this cell from this cell and look deep within inside it, we'll find gap junctions there. Okay, so again, the heart is involuntary, it's striated, it's highly branched, only found on the heart. Smooth, this one looks spindle-shaped, right? They are spindle-shaped, they're flatter, you've got central nuclei. Look, they're closely packed together here in smooth muscle. These are around the organs like the uterus, the esophagus, the stomach, the colon, the small intestine, a woman's uterus, your blood vessels. In fact, when I do the histology video, when you watch that one later, you will see that there is one that shows an artery. Okay, and the artery is perfect because an artery, the layer that's closest to the lumen is epithelium. It's squamous cell epithelium. Squamous, simple squamous, in fact, because it's only one cell layer thick. That's closest to the lumen. Then deep to that is smooth muscle, and it is that smooth muscle of an artery that makes it constrict or dilate. If an artery constricts, blood pressure goes up. If the smooth muscle dilates and opens up, blood pressure goes down. So an artery, the innermost part closest to the lumen, is one cell layer thick of squamous cells, so it's simple squamous epithelium. Then external to that, you're going to have smooth muscle. And then what's in the lumen itself is blood. That's liquid connective tissue. So you have a little bit of both. You've got red blood cells, which is liquid connective tissue. You have simple squamous epithelium, and then you have muscle, right? So you got three different types of the tissues of the four. That's pretty neat. Okay, and then last, we have the neuron. So we finished epithelium, connective, and we did muscle, the three types. Last but not least, we have the neuron. And this is where I opened up today, and I said, if you look at your hand, your palm and your forearm. Here, these would be your fingertips, this would be your palm, and this would be your forearm. So here, these are the dendrites, which comes from the Greek word dendra, which means branches of a tree. So these are dendrites. They're bringing in information into the cell body or the soma. Soma or cell body, it's the same thing. So dendrites bring in neurological sensory input into the cell body. Then the soma or the cell body processes it and sends the information away through the axon. So just remember the A in axon for away, A and away. So information comes to the cell body or soma through the dendrites, and then the electrical impulses move away through the axon. One of the things to pay attention to is that the axon is covered by myelin. Myelin is fat. It is fat, it's made up of fat, it's made up of cholesterol. It is basically a insulated protection where if you were to look at any electrical cord, you'll see that you don't see the electrical wires to any electrical cord. It is fully protected, right? If you were to touch what's deep to that insulation, whether the wire was, the copper wires were insulated by something black or it was insulated by something white, still the covering is designed to speed up the electrical conduction. So this myelin sheath is designed to speed up electrical conduction. It's going to send the signal all the way to the very end of the axon, all the way at the end of the axon. They're called axon terminals. Just remember, like when you go to the airport to catch the plane at the end, it's the terminal. You got to go to the terminal, the end, the axon terminal. And at the very end of the axon terminals are end bulbs. And what's contained at the very end of the end bulbs are neurotransmitters. Neurotransmitters are stored here. And then when the body needs to, it releases them. And some neurotransmitters are excitatory. It's like an on switch. And some are inhibitory. It's an off switch, depending on whether we need to send a chemical message that says turn on or turn off. So neurotransmitters could be stimulatory or they could be inhibitory. Okay. So, like, for example, when you get up in the morning and your brain turns on and you need to pay attention, that's your body sending out glutamate. That's a stimulatory neurotransmitter in the brain. But then glutamate, by the time it's 7, 8, 9 o'clock at night, turns into GABA. GABA is a very inhibitory neurotransmitter that's calming to the brain and it helps you sleep at night. So some neurotransmitters get you up and then some neurotransmitters turn you off. All right. So those are the main parts of the neuron: the dendrites, the cell body, the axon, and the axon terminal. The axon is covered by myelin. When you get into neurology, you will learn about one of the or two neuroglial cells that produce myelin. One of them is called a Schwann cell, Schwann, Schwann cell, that produces myelin in the peripheral neural system, whereas the oligodendrocyte, an oligodendrocyte produces myelin in the central neural system. Central neural system meaning the brain and the spinal cord only. Anything outside of the brain, like your cranial nerves, anything outside of your spinal cord, like your spinal nerves, or your sciatic nerve and your median nerve and your radial nerve, all of the peripheral nerves, those are going to be myelinated by Schwann cells. Each of these is a Schwann cell, and they produce myelin to speed up the electrical conduction, to increase the speed and velocity of the action potentials or electrical conduction or nerve impulses to make it to the very, very end. Okay, so make sure that you look at the histology slides in a separate video, the PowerPoint file, and the video that goes over all of those um, s- different types of epithelium, connective, muscle, and nerve that we just covered. Okay, let's stop the screen share here and let's end the video recording.